Right. Okay. So the three types of cells. Generally, you have the squamous cell, cuboid cell, and the columnar cell. Squamous cell is designed for diffusion, cuboidal and columnar cells for absorption and secretion. Okay. So you can't see the cell margins through microscopy. So you have to, you generally look at the size and the shape of the nucleus, and then that becomes a guide to your cell size and shape. Okay, so if you see a flat nucleus which is sort of bulging out a little bit, but it is parallel to the basement membrane, then it is likely that it is a squamous cell, right? And a cuboidal cell generally has a rounded nucleus in the center of the cell. Okay, and it is the same distance from the lumen and from the basement membrane. So it is placed centrally in the cytoplasm. Then the columnar cell, if you look at, it's usually an elongated, ovoid-shaped nucleus. Generally, the nucleus is closer to the base than to the luminal surface. Okay. Right, so this is a high magnification, 10 to the power 40 of lung tissue, right? So surrounding here, the white areas, that is air. So this is the alveolar air. Okay, all this is air, and this is the cell, right? And this is also again another cell, and that is the nucleus. Okay, so that is what you know the shape of alveolus. It will be shaped like this, right? So three-dimensional, if you look at it, it is like this. So you have the alveolus being lined by all these squamous cells, right? So when you nucleus will be somewhat flattened like this. Okay, so when you take a high power view of the lung, then this section has been taken and you can see those two cells there, right? So like that, you see the two cells with their flattened nuclei. Okay, so observe the squamous cells in the lung tissue slide. This is a Cheney section. The nucleus is thin and flat with a small amount of cytoplasm, right? So that the cell is designed to allow substances to diffuse through it, right? So things can get through the cytoplasm of these cells in and out easily, right? That doesn't mean everything will come out and get in. There will, of course, be some sort of a protection against any invading organisms. The dust particles, large particles cannot get through, right? So for diffusion to occur, molecules have to be small. Okay, so water vapor, then gases like oxygen and carbon dioxide, which are small molecules, can go through the cytoplasm of these cells, right? So since it's epithelium, nothing is allowed to pass between the cells. Everything has to go through the cytoplasm. Okay, right. Okay, so then this is a cuboidal cell. This is basically a thyroid tissue, right? So again, high magnification, 10 to the power 40, thyroid tissue. Okay, so this is thyroid. So this would be colloid. The pink material inside is colloid, and surrounding the colloid, you have a line of cells. So the basement membrane would be somewhere there, and you have a line of cells. Okay, and you can see that the nucleus is rounded. Okay, so you know the thyroid gland, which is in your neck, right? So it's like a butterfly shaped gland in your neck. Okay, so if you take a cross section of it and look at it, it will have these, right? So in endocrine gland lectures, exocrine gland lecture I showed you, sorry, glandular classification lecture I showed you, right? So in the center, and the cells are cuboidal, single layer of cuboidal cells with central rounded nuclei. Okay, so the round nucleus is placed at the center of the cell, and a considerable amount of cytoplasm is there, right? Considerable amount, because it's quite a, I mean, it is a bit of a larger cell. Okay, so central rounded nucleus. Okay, right. Then columnar cell. Here you can see one columnar cell, right? So that is the basal surface, and this area is called the lamina propria. Okay, so these are connective tissue, that is the lamina propria. Okay. It has connective tissue, right? Okay, so it has connective tissue and all sorts of cells there, right? Different immune cells and things. And these lines are connective tissue, right? And here you get the basement membrane, and on top you have the columnar cells, right? And you can see that the nucleus is oval shaped or elongated, and the distance between the nucleus and the base is less than the distance between the lumen, right? So cell would be like this, basement membrane like this, and the nucleus would be like this, right? So the distance here is less than the distance here. Okay, so observe the columnar cells with their elongated nuclei placed closer to the basal surface of the cell, right? Here, of course, you can't see the brush border, but you would get microvilli somewhere there. Okay, so in some slides, you will be able to see the microvilli, the brush border. Okay, right? So then going on to membrane specializations, which we discussed. Now, some luminal surface, some cells in the luminal surface of epithelia can have specializations as we discussed. Okay, so cilia, microvilli, or stereocilia. Okay, right? So if you remember this slide, I don't know whether I showed this to you, but this is projections from the luminal surface of the cells. It is basically the cell membrane just projecting up like this. Okay, so the cell will be like this with the nucleus, right? So this epithelium is pseudostratified columnar ciliated, that will be the basement membrane, and these are the cilia. Okay, so they are quite large and you can see them quite clearly. Okay, so those are all cilia, and as you know, these cilia are motile. They can beat like a wave, right? They have the ability to move, and because of that, if there is mucus on top, that mucus will be propelled on the surface. Okay, so this is actually epithelium from the lining of the nose, right? So these are actually glands, mucus secreting glands, which you see there. Okay, and this is a pseudostratified columnar ciliated epithelium, which we call the respiratory epithelium. Okay, so this is actually from the nasal passage, right, wall of the nose. Okay, right, so cilia in the epithelium of the lining of the nasal cavity. Okay, right. Okay, and then microvilli here, of course, because this is a very high power view, I think this is 10 to the power 60. Okay, high power view. You can see the finger-like projections of the plasma membrane. Sorry, what did I say? Okay, so here you have the membranes coming like this, and in the center you have the filaments, right? So you have the filaments like this. Okay, so it has a skeleton, and that is why it is motile. Okay, and there's a base also where it is stuck to the base like this, right? So because of the specific structure with several filaments going up the cytoplasm lining, the cilia has motility. Okay, so you have the filaments in the middle, and also they are quite long. Okay, they're quite long, right? Now this is very high power, 10 to the power 60, and you have finger-like projections of the cytoplasm, but you don't have the filaments. Okay, so you don't have this base or filament, and these are non-motile. They cannot move. Okay, so these are just there to increase the surface area of these columnar cells, right? And we call them the brush border. So this is the brush border of the interstitial intestinal epithelium, right? Sorry, intestinal epithelium. This is the brush border. Now here you can see only a hazy appearance, right? This is sort of a hazy appearance on top of the columnar cells. Okay, and this one here, this is a goblet cell. Okay, and these are they look like sort of crowded like this. Okay, all right, but actually they are in the same plane. Okay, though they look a bit crowded. Okay, so this is the wall of the small intestine where you can see simple columnar epithelium with microvilli, right? So observe the hazy appearance on the luminous surface of the columnar cells, right? Okay, so this is again HNE, okay, 10 to the power 60, small intestine. Right? Okay, so stereocilia, as I told you, these are not mobile, right? I'm not really absolutely sure. You can read up some books also and see. So they are said to be long microvilli, right? So they are just projections of the cell membrane. Okay. All right. So then this is again a pseudostratified columnar epithelium with these long microvilli extending into the lumen. Okay. Right. So then going on to the classification, I think we've gone through this. You know this very well. So you have the simple epithelia and the stratified epithelia. And we'll talk about simple squamous, simple cuboidal, simple columnar ciliated, pseudostratified columnar ciliated, and the stratified squamous, two types, stratified cuboidal and columnar, and the transitional epithelium, right? Okay. Right. So again, to look at the simple squamous epithelium, the best thing is to look at the lung alveolar wall, right? So again, this is a high power view, 10 to the power 40, and actually this should be 10 to the power 10, I think, right? So 10 to the power 10, okay? And you can see a single lining epithelium of the alveoli, right? So note the single layer of cells forming the walls of the alveolus. This is an alveolus, one alveolus. Okay, so this is 10 to the power 10 because this section has been taken separately. It looks large, but it's 10 to the power 10, right? And these are the small cells. You can just make out the nuclei. Okay, so because it's a low power view, you can't really see the nuclei that well, right? So the thin wall of the alveolar wall facilitates diffusion of gases, right? And you can see the flattened nuclei and the tiny amount of cytoplasm in the squamous cells, right? Okay, and then here, this is a blood vessel, right? So here these are the red blood cells inside this RBCs. Okay, and here you can see some white blood cells. Okay, so the red dots are the red blood cells, and these nuclear ones are the white blood cells, right? So surrounding that, you can see the wall of the blood vessel. Okay, so you can see flattened nuclei, right? Okay, so this is high power view, 10 to the power 10 HNE blood vessel, right? So we call it simple squamous epithelium is called, when it lines a blood vessel, is called an endothelium, right? So the lining epithelium of the blood vessels, lymphatics. Blood vessels means arterial system, venous system, and the lymphatics. We call them endothelium. Okay. Right, so here you can see smaller blood vessels. This is probably a capillary. Okay, so these are capillaries. Now here you can see very thin, small amount of connective tissue surrounding it, right? So these are small blood vessels. Yeah, it's easier for you to see the wall. Thick blood vessels, every blood vessels and the heart are lined by simple squamous epithelium, right? Okay, so again, simple cuboidal epithelium, as we looked at previously, you see it in the thyroid, right? So rounded nucleus, and here these are the follicular cells, right? And this is the interfollicular spaces where you have the connective tissue and small cross sections of blood vessels, right? So this is the colloid, as I told you earlier, and the simple cuboidal cells. Okay, right? So this is again 10 to the power 40 H&E section of the thyroid tissue. Okay. Right, so this is actually simple cuboidal epithelium of a duct in the salivary gland. This is 10 to the power 40 or maybe it's 60, not really sure. Maybe 60 also, right? Okay, so here you can see very nicely the nuclei, right? So this is a nucleus, and surrounding it you can see the cytoplasm, and you can imagine the cell margin. Okay, so the basement membrane would be on this side like this. That is the basement membrane, and that is the lumen of the duct. Okay, so whatever secretions comes through the duct will be traveling in this lumen. Okay, so you can see this very nicely, and you can see the nuclear margin. You can't see it, you can just imagine where it is, and you can see the chromatin, right? You can see the granular appearance of the chromatin. Okay, purple stained nucleus. Okay, right? So note the single layer of cuboidal cells lining the lumen of the duct. Observe the rounded nuclei placed in a line surrounding the lumen of the duct. Okay. Right. So then simple columnar epithelium, as we saw in the small intestine, whole of the intestine starting from the stomach to the rectum are lined by simple columnar epithelium. There is no other epithelium in the gut, right? So you get only the esophagus and the oral cavity do you get a stratified squamous non-keratinizing epithelium, right? Esophagus, and in the oral cavity, of course, you might get keratinized epithelium in some areas. Okay, right. So then the tallest columnar cells are seen in the small intestine. If you take the stomach epithelium, slightly less tall, right? So the tallest ones are seen in the small intestine. Okay, and when you take a longitudinal section, they appear columnar in section, and the nuclei are elongated, as I told you, situated towards the base, and of course you can see the goblet cells, right? So goblet cells are specialized cells. They are glandular cells, and they are called single celled glands, right? So these are the goblet cells, right? So they are called goblet cells because they look like a goblet, wine goblet, right? Like that. Okay, that would be a wine goblet, right? So goblet cells generally look like that. Okay, so they're like this, and the cytoplasm is going down like this. So the nucleus will be somewhere here, and all this is filled with mucus, right? So the cells are filled with mucus. As I always say, mucus doesn't stain well with H&E, right? Okay, so this here is a section from the fallopian tube, right? The uterine tube, and a uterine tube is convoluted. That is why it looks like this when you cut across it. It has a single layer of columnar cells, right? Simple columnar epithelium. However, it has cilia on the luminal surface, right? So you can see the cilia here. Now, different ciliary epithelia may be slightly different. Okay, so in the respiratory epithelium, all the cilia of the same length, right? And they look very sort of neat and all that. But in the fallopian tube, it may be a little bit different, right? However, all these are cilia. Okay? So the cilia will help, they will beat in one side. So cilia in the fallopian tube will beat towards the body of the uterine cavity. Okay, and then they help the ovum to travel along the tube. Okay. Okay, so again, this is H and E section, fallopian tube, 10 to the power 40. Okay. Right, so the next slide is, these are all the slides that we have in the lab, right? So we have taken photographs and we're just showing it to you so that if you get a chance to come and look it through the microscope, you know what you're looking for, right? Looking at, and so you won't get the same amount of time to study this. That's why we've done this online, so that you can study it here, and later you will be given, you will have only very small amount of time to actually go through, because you won't have time, right? So when you come to the faculty, it will be for a short duration. Okay, right. Okay, so here you see pseudostratified columnar ciliated epithelium, which, as I said, is the respiratory epithelium, and it is a simple epithelium because all of these cells are actually lining or lying on the basement membrane. Okay, so these nuclei appear as if they are in different levels because the cells are of different sizes. Okay, right, and that is why it appears stratified. It is not a stratified epithelium, it's a simple epithelium. Okay, and it lines the nasal cavity, trachea, and the bronchi. All right, so this is again H&E 10 to the power 40 high magnification slide. So below the basement membrane, this here you can see the connective tissue and cross sections of blood vessels and some cells, immune cells, mostly lymphocytes, right? So these are lymphocytes. Okay, and here you can see the cilia. These are generally of the same size, like. Okay, and on top of this, you would get a layer of mucus. Okay, then moving on to the stratified epithelia, you have more than one layer of cells. The basal layer is on the basement membrane, and according to the shape of the cells, right? So you have stratified squamous non-keratinizing and keratinized, and stratified cuboidal columnar, and also the special epithelium, which we call transitional. Okay, right? So here you see stratified squamous non-keratinizing epithelium slide. This is a slide from the esophagus, right? So the esophagus and places in the oral cavity are lined by this epithelium. This is again 10 to the power 40 H&E section of the esophagus. Okay, so you can see several layers of cells are there, right? And the basal cell layer is cuboidal in shape, lying on the basement membrane, and the top cell layer is squamous shaped. Okay, so that's why we call it a stratified squamous cell, and there is no keratin on the surface, so we call it non-keratinizing epithelium, right? Non-keratinizing epithelium, because there is no layer of keratin on top. Okay, right? So these cells are cuboidal in shape, and these are polygonal, right? Polygonal because they are getting compressed from all sides. Right? Okay, so then again, stratified squamous non-keratinizing epithelium. This is a section from the vagina. Okay, so again, H&E 10 to the power 40 high magnification. So you can see the basal cell layer, which have cuboidal shape, right? And the basement membrane and the connective tissue here, and then several layers of cells. Okay, as it goes up, you can see that the cells are becoming more and more flat, and you can see all the nuclei up to the topmost layer. You can see the nuclei. Okay, and the top layer is lined by squamous, simple squamous cells. Okay, right. So then coming on to stratified squamous keratinizing epithelium. you can see that there is a layer of keratin on the top, right? So thick skin, right? Thick skin. This is thick skin where you have more layers. This corneum lucidum, the lucid layer is there in thick skin, right? And here you can see the dermal papillae here, right? And you can see the basement membrane will be there, and the basal cell layer and the other cell layers. Okay, so the topmost one is the keratinized layer, right? And a keratin layer, and then the stratum corneum. Okay, right? So layers of keratin above the superficial layer of cells, making this a keratinizing epithelium, right? So the cells in the skin produce the hormone keratin, and the cell cytoplasm gets filled with keratin, and as it gets towards the surface, the organelles are extruded, right? And the cell actually dies. So you have only a cell membrane, and inside the cytoplasm is filled with keratin. And finally, those cells can be shed. Okay, and all the time these basal cells divide and replace the cells that are shed. Okay, so this is of course a low power view so that you can see a wider range, right? So 10 to the power 10 H&E section. Then coming to the transitional epithelium, so you can see a special type of stratified epithelium which is limited to the urinary system, right? So it lines the pelvis, the pelvic axial system of the kidney, the ureter and the bladder, and some parts of the urethra. Okay, so this is designed to withstand the stress, and this is H&E section 10 to the power 40, and you can see the scalloped edge, right? So the lining cells of this epithelium, we call them, they are the luminal surface of these cells are impermeable to water and urine, and we call them umbrella cells or domed cells. And you can see their nuclei and several layers of cells, and then beneath that the connective tissues, right? And these are cross sections of blood vessels. Okay, so topmost layer is umbrella cells. Okay, right? So then going on to glands, you discussed about glands a great deal, right? And you have sent me your assignments. I think most of your answers, almost 90% of the answers are extremely well written, nicely structured and all that. So I have given you comments as well. There were few which didn't have enough material. So I have mentioned that also. So you can just develop a little bit more on it. So yeah, so the answers are all good, right? Okay, so then endocrine glands and exocrine glands. Endocrine glands don't have ducts. Exocrine glands have ducts, right? And they secrete to the surface of the epithelium. Okay, so then now some of you had missed these small things. Now to add to that, if you just talk about serous glands, mucous glands, mixed glands, and also how you define glands by the secretory type, like merocrine, holocrine, and the other one, yeah, those three types of modes of secretion, right? So if you talk about that, then that also will add to your answer, right? But if you explain how the multicellular type of exocrine gland classification, then you will get most of the marks, right? About 65 marks you will get from that. If you have added about single cellular glands, multicellular glands, and also this part about serous, mucous, and mixed glands, then you will get definitely close to 75 marks. If you added all of that together and organized your answer really nicely and all that, then you get full marks, right? So some of you had drawn diagrams also. Even though diagram is not asked for, if you do draw, then it shows the examiner that you have a good idea about it, right? So it's good. But if it is mentioned that you have to do it, if it says giving examples and using diagrams to illustrate your answer, then you have to draw diagrams, right? If you don't draw diagrams, you won't get marks. But if it is not mentioned in the question, then particular mark is not separately given to the diagrams, but it will add to your answer. Okay, right. Okay, so you have this, you know, I told you about this serous, mucus and mixed glands. Okay, so serous glands, as you've seen this, so this is a slide that we have of the pancreas, right? So this is 10 to the power 40. It is focusing on the areas where, because the pancreas is it's sort of, it has endocrine and exocrine functions both, but here you see the exocrine one, right? So these are the acini, okay, of the pancreas with the tiny lumen in the middle. This is a blood vessel cross section. These are red blood cells. Okay, so acini are lined by acinar cells. Acini have rounded nuclei. They are darkly stained because of the cytoplasm has a lot of secretory vesicles, which is full of enzymes basically. So they are made out of proteins. And you can see cross sections of the blood vessels because, of course, pancreas has a lot of blood vessels because it has the endocrine function as well. Okay. Right. So then mucous glands, you have these lightly staining ones with large mucous granules and the peripheral nucleus, right? So the lumen is there, the cell is there. You have a large mucous vesicle here and a peripheral nucleus, right? So the nucleus is pushed to the periphery. Okay. Right? Note the acini lined with acinar cells. Acinar cells are filled with mucous secretions. Cytoplasm of the acinar cells are stained poorly because the mucus does not stain well with H&E. Right? And the nucleus is flattened and pushed to the periphery due to the mucus. Okay. Right? Then mixed glands you have. So this is a gland, the slide of the salivary gland. Okay. Okay, so here you have both mucus and the serous acini, and the serous acini are darkly stained, as I've told you many times. Mucous acini are lightly stained, and you can see the serous demilunes, right? Serous demilunes, mucus cells, mucus acini, serous acini, and these are, of course, the ducts, right? Okay, so serous demilunes appear like a crescent. That means demilune means crescent, half moon shaped. Okay. Right. So then again, the classification. Now I'm doing the lecture, then I'm doing this again for you. You have done your questions. So just, I think that is more than enough for you to remember your foundation, initial tissue things. You don't have to go through this again. It should be enough, right? So you can put that away and concentrate on your limbs, the limbs lectures, which one has already been done. So from next week, okay, and we will show you dissection videos and all that. So you can concentrate on that and start to read your Snell, right, on limb sections. So it would be really useful if you can read it a little bit before you go for the lecture, right? So on Monday, I don't think you have got the timetable, most probably not. You will get a lecture on the axilla and the brachial plexus, right? Axilla and the brachial plexus on Monday. So if you can read that section in your Snell before you come for the lecture during the weekend, it will be good for you, right? So the limbs has a lot of things that you haven't really done yet. Now, these are things that you have done in A-levels. So quickly get this into your head and keep them aside so that it'll be there when you want it for the exam, right? So, and then concentrate on mainly understanding the structure of the limbs. Okay, right. Okay, so I'm not going to go on and on about this. You have the simple glands and the compound glands. We'll just look at the slides of each of these, right? So simple acinar gland, this is, as I said, this cup-like thing. So there is a small, maybe a small, you can't really see whether which is which, right? So there's a small duct and ends with the acinar portion. Okay, so this is a simple acinar gland. Okay. Then simple tubular glands are quite common, right? So you see them in the stomach, fundus and body of the stomach, and also in the colon, right? So the duct, I mean, you can't really see which part is the duct and which part is the gland. It's difficult, right? The whole thing just looks like a tube like this. Okay. However, we call part of it the duct and the other part the secretory portion. However, all this is lined by mucus cells, right? So the secretory part is tubular shaped. The duct also continues with that tubular shape. All right. So the colon epithelium, whole of it is lined by these simple tubular glands, right? So this is an HNE section, 10 to the power 40, the colon. So these are also called straight tubular glands. Straight tubular glands. So if you say straight tubular glands or simple tubular glands, it basically means the same thing, right? Okay, and then coming to simple branched tubular glands, where the duct isn't branched, but you can see that these secretory portions are branched. That's why you have these cross sections where you have circular areas. So if you take the duct which is like this and cut across, cut across like this, okay, cut across like this, then you will see some parts of it getting cut as circles. Okay, so that is because of the branching. So if you cut across like this, you get some will be circular like this, others will be long like this, right? So the duct will come down and the tubular portion will be branched, right? So the duct is not branched, but the gland is secretory component is branched. Okay, so you see this in the pyloric part of the stomach. This is actually because the pyloric part of the stomach, after that you get the duodenum, right? So the stomach is full of acid, and it is this mucus that prevents the acid from going into the tissues inside and sort of destroying them, right? So the stomach, the acidic contents of the stomach has to become basic so that they can get into the duodenum, because the duodenum is not designed to withstand a lot of acidic content, right? So if a lot of acidic content gets into the duodenum can get damaged. You can get ulcers in the duodenum, right? So because of that, lots of mucus is secreted in the pyloric region. So these simple tubular glands become simple branch tubular glands when you come to the pylorus, so that more and more mucus can be secreted, right? So this is directly related to the function. Okay. Right, so then simple branched acinar glands, where you have again the duct is not branched, but the secretory part or the acinar part of it is branched. Okay, so the sebaceous glands in the skin is an example. As we saw, sebaceous glands are there. Most of the time, the sebaceous glands opens into a hair follicle, side of a hair follicle. So hair follicle is like this, and to the side you get the opening of the sebaceous gland, right? So singly lying ones can also be there like this one. So this is the duct. Okay, so this is a special stain. Okay, so this is actually 10 to the power 10 special stain. Special stain skin. Okay, so this is thin skin, right? So you get hair follicles in thin skin, right? On the scalp and on the body, you get thin skin where you get hair follicles and sebaceous glands, right? So most of your sebaceous glands are actually in your scalp. Lots of sebaceous glands are there in the scalp. Other areas also you get sebaceous glands. But in the palms and the soles, you don't get hair, right? So in the thick skin, there are no hair follicles. Okay. Right, so the duct is not branched, secretory acinar portions are branched, and sebaceous glands of the skin. Okay. Right, so here you can see the acinar cells inside with the nuclei, right? a little not very clear because this is a low power view to show you the whole gland. Right? Then simple coil tubular glands. These are glandular parts of the sweat gland. Right? So I showed you a picture that looks like this, but it is very difficult to get a section like that. You have to cut it exactly from there. Right? And when you cut it across like this, from several different areas only, this gets cut, right? So you have the duct and the acini. Okay, so the duct is not branched, the tibular secretory part is coiled, and the secretory portion is lined by simple cuboidal epithelium. Okay, and the duct is lined by stratified cuboidal epithelium. Because of that, when you see these circles, you know that this is a duct. Okay, this is a duct, this is a duct, these are acina portions. Okay, so like that, and these of course connective tissue surrounding it. This is again a skin special stain. This is 10 to the power 60. Okay, so simple coil tubular gland is specific for sweat glands. You don't see it anywhere else in the human body, right? So sweat glands are also of different types, and they are present in all the skin, right? Okay, and then this is the Brunner's glands that we talked about. So compound branch tubular gland. We have this slide. Now this is a very sort of important slide because you can see the duct here, right? It's very difficult to find a duct like this opening into the epithelial surface, right? So the slides with ducts are difficult. Okay, so you can see this is the muscularis mucosae, muscle layer of the mucosa, and you can see that it is not there in this area. Okay, so the gland is there, that is the gland. Okay, and that is the duct. All right, so you don't really see the branching. Here you can see a little bit of the branching of the duct as well. Okay, so the duct and the tubular secretory parts are both branched. Okay, so this is a compound branched tubular gland, and that is the Brunner's gland. That is actually the largest type of gland which can secrete the most amount of mucus. Okay, so as I said, when it comes to when the food gets mixed and comes into the duodenum has to withstand that acid and quickly make it basic, so that lots of mucus can get secreted onto the surface of the duodenum and increase the pH of the food that is coming in. Okay. Okay, so those are the slides we have of glands. Next, we have a few slides to show you the connective tissues, right? Right, so classification you have already done. Ordinary connective tissue, cartilage, bone, and blood. So we'll be talking about ordinary connective tissue. We will do the cartilage and bone later. Okay, so ordinary connective tissue, as you probably did in your lecture, you have several different types. So we will look at slides of loose irregular tissue, dense irregular, dense regular, and adipose tissues. Okay. Right, so this is loose irregular connective tissue. Collagen fibers are arranged in irregular manner, right? So you can see scattered fibroblasts between the collagen fibers, small blood vessels, cross sections, and this kind of thing is actually found in the lamina propria and in the deeper layers of the skin. So you saw the lamina propria in the small intestine and all the gut areas, and also just beneath any epithelium in organs, you get this loosely arranged connective tissue, which acts like a packing material or filling of the gaps, and they allow blood vessels to go close to the epithelia, right? So this is really important for the nourishment of the epithelia. Okay, so this is this here is the cross section of blood vessels. You can see the red blood cells inside. Then the collagen fibers stained in pink, irregularly arranged, right? And the small fibroblasts. Okay, so you know different types of cells are there in the connective tissue. Fibroblasts are easy to see. Right, so then dense irregular connective tissue is seen in skin. Now this is a Mason's trichrome. Just remember, it's a special stain where collagen is stained in blue, right? So this is a special stain for collagen. Usually H&E collagen stains in light pink and it's difficult to see. But because you want to look at these densely arranged collagen fibers, you stain it in this special stain. Right, so this is a low power view of a thick skin showing you the collagen fibers and how they are arranged. Right, so you can see densely packed, irregularly arranged collagen fibers stained in blue. Okay, so here, of course, this is a part of a sebaceous gland, and this is muscle. This is the epithelium, epidermis, and that is the dermis. Right, so you can see lots of collagen fibers, but they are arranged in an irregular manner. Okay. Then, of course, you have dense regular connective tissues in some capsules and also in ligaments and in joint capsules, ligaments, tendons, aponeurosis. You see the dense regular connective tissue, which is the most strong connective tissue that you can have, right? So it covers organs, it covers and contains the joints, and also it gives attachments to muscles and connects bones together through tendons and ligaments and aponeurosis, right? So aponeurosis is actually when the muscle is there, all of these even the muscle is covered by layers of connective tissue, right? And these connective tissue coming together will form aponeurosis, and also at the ends of each of the muscle will generally end with a tendon, right? So the tendon is dense regular connective tissue, and it is the tendon which gets attached to the bone, right? So you can see here the capsule of the testis, which has regularly arranged collagen tissues very tightly packed together, right? Okay, so then the last slide where we see the adipose tissues. Now again, adipose tissues, the adipocytes are, cytoplasm is completely filled up of a large fat droplet and the nucleus is pushed to the periphery. Okay, and what happens here is when you are processing the slides, you saw that we put alcohol, right? So when you put alcohol, the fat inside these cells gets dissolved and the empty space is only left. Okay, and the cytoplasm gets pushed to the periphery and you get the thin pink stain with eosine, and you can see sort of like empty spaces, like, so all this is fat, that is the nucleus, and thin rims of cytoplasm are seen. Okay, so this is what a adipose tissue will look like, and adipose tissue is found right underneath your skin. So the hypodermis of the skin is made out of fat, right? So this adipose tissue, and underneath that also you get the subcutaneous fat underneath the skin, and also surrounding organs within body cavities like the peritoneal cavity. In the abdominal cavity, you get lots of fat being deposited. Okay, so it is not subcutaneous fat, it is the fat which, it is a subcutaneous fat as well as the fat which is inside the abdomen, in the peritoneum and also surrounding organs, right? So all that is made out of adipose tissue. Okay, so that is all. Now I will take questions. We have a few minutes. Yeah, so I can take questions. If you have questions, you can type on the chat box, right? Or this lecture, this practical session or any lectures that we have done up to now. You can, if you have any questions, you can type. Yes, so we are going to give you slides, pictures of microscopic view. So if somebody gives you this picture, if you may be asked, identify the tissue. So you will write adipose tissue. And then the second question may be, give reasons for your identification. Then you will say, these are cells filled with a lipid droplet, which is not stained. There's a peripheral thin rim of cytoplasm and a peripheral nucleus. So this is adipose tissue. And then you might ask, what is the function of this tissue? So you will say, it is found in subcutaneous, it is a storage of fat. It also helps in temperature regulation, and it helps to protect different organs, something like that, right? Okay, then what are the places you can observe keratinized stratified epithelium except thick skin? Yeah, so keratinized epithelium is, you can see some parts of the oral cavity, right? Like the hard palate, okay, hard palate, and areas of the tongue, and some areas of the mucosa where you get the teeth, right? The mandible and the maxilla where you get the mucous membrane. Some parts of that may be keratinized, okay? Otherwise, it's the skin. The magnification, not really. So do we have to find the magnification? Not really. You won't be asked whether it's high magnification and low magnification, maybe, but not exactly whether it's 40 or 10 or 4, that kind of thing, generally not asked, okay? How communicating junctions control the growth of the cell? No, the thing is, growth of the cells means growth factors, right? So growth factors may be secreted by different cells, different other cells, or brought from the blood. So if it comes into one cell, through the communicating junctions, those molecules will go to each of the cells, right? So that all the cells can grow together. Otherwise, you will have all sorts of cells growing at different rates, and then it won't be properly maintained, right? So communicating junctions actually help cells to know what the other cell is doing. Endosteum cover trabeculae of cancellous bone. Yes, they do, right? Endosteum covers it. Meaning of mucociliary escalator. Okay, so you have, I'm sure you have seen escalator. That means something moving up or down in one direction, right? So when you say mucociliary escalator, basically it means the cilia are beating in a way towards the upper airways, carrying the mucus up with it, right? Like people being carried up in an escalator. So if there's a dust particle or a foreign body, it gets trapped in the mucus, it gets pushed upwards, right? So that is what we call a mucociliary escalator. Mucus and the cilia together acting like an escalator to push foreign bodies or dust particles up towards the upper airways, okay? Both cilia and microvilli in simple columnar epithelium. Yes, simple columnar epithelium, yes. You can find microvilli in the gut. You can find cilia in simple columnar epithelium in the fallopian tube. Yes, correct. Cilia and microvilli. Yeah, I know this is, I mean, it might seem like difficult, but cilia are actually quite easily visible, right? Cilia are easily visible. You can see that they're quite long and they're easily seen, but microvilli, you can't really see them as lines, only like a hazy appearance, right? So, so it's not that difficult to differentiate, okay? Just see this. Okay, so here you can see them, they are like lines, right? Lines like things, but if this has a sort of a hazy appearance, right? So that is the brush board or the microvilli. They're very hazy. Okay, you can't really see them as lines, but here you can see them like lines, right? So it may seem difficult, but actually when you look at it, it is not that difficult, right? And also by the other features, you know that what is what. Okay. How do I find whether it's low or high magnification? By the things that you can see, right? So if you can see the wide range and smaller nuclei, smaller structures, that means low power. If it is the cells look really large, then it is high power. Okay. Actually, lecture notes are more than enough to write your essays, but you can read a little bit surrounding it to make you remember more, and also you can add to those things, right? If you write exactly what is there in your lecture note, you will definitely get more than 65 marks, right? But if you want to, improve more, you might read up a little bit more, okay? But these lectures, of course, more than enough. You don't have to go into too much details. Are there cuboidal epithelium, microvilli are there in the proximal convoluted tubule, right? Proximal convoluted tubule of the nephron has tall cuboidal cells with microvilli. Okay, yes. Yes, stereocilia found only in the epididymis and the vas difference, right? Yes, correct. Polarity of the simple columnar epithelium. Basically, all epithelial cells have polarity, right? Not only simple columnar. Polarity means the cell has two different sides. Now, if you take a fibroblast or a blood cell or a macrophage, they don't have polarity. Okay, the whole of the whole cell is the same, right? However, if you take a hepatocyte, they don't have polarity, right? So you have epithelial cells have polarity because one side is adhered to the basement membrane and the other side is luminal. Okay, so even if you take the epithelial cells out of that basement membrane, still that polarity is there. Okay, so one side is designated to be attached to the basement membrane, opposite side is designated to be on the luminal side. That's what it means, right? Stereocilia and cilia. Yeah, okay. So again, cilia, as I told you, same sort of length, and they're shorter, and or like whole of the luminal surface is full of cilia. Stereocilia, only the gap between each of the cilia are a little bit more, right? And they're longer. Okay. Yeah, below the tight junctions, yeah, only in some cells do you get this whole addition belts and all that, right? The complex is there only in some cells, but you get tight junctions you get in all epithelial cells, right? And zonular adherence also, right? Especially you see this in the gut, right? Intestinal epithelium. No, stereocilia can't move. Microvilli are not there in goblet cells. Okay, so the limitation in answering questions when you ask to describe or explain. Actually, there is no limit. It is basically whatever you can write within that time period that is given to you, either 15 minutes or 20 minutes, according to that. So whatever you can remember, you will write. Now, when you're writing assignment, you are looking at the books, looking at the lecture, and then writing. But at the exam, you will write what you can remember, right? So you write down everything that you can remember in the given time. Always remember, don't exceed the time, stick to the point. Don't write something that is out of the point, right? So if you're asked to write the classification of exocrine glands, you don't have to say a word even about endocrine glands, okay? You don't even actually have to define what a gland is, but having that is okay if you have time. But you will not be, marks won't be cut, but you will lose time if you write things which are not necessary. Okay, so marking schemes are very specific. We have marking schemes have points. Okay, points are there, and for each point you will get a mark, and then organizing and designing the answer, you will get again some points, but mostly it will be for each of the things that you have written, right? So when you say describe, That's what I have told you in your answers, to write a little extra, right? So it is the way you write it. You can actually, if somebody says outline or list, okay, it will be different. List the different types of glands. If you are told that, you just go on writing simple acinar gland, simple tubular gland, like that. If you are told to describe it, then you have to say a little bit more detail, right? So explain is different. Explain is actually given to a sort of, if you're asking about a clinical correlation or why something happens, then you have to explain why that happens, right? Okay, so and also you can take a guideline by the number of marks that is allocated, right? So if it is a 100-mark question, you write the maximum. If it is 50-mark question, then you can decide how you're going to approach it and write it. Okay. A bit more detail about alpha and beta keratin. I don't think so. I'm not sure really. Is it, you can look at your biochemistry notes. Probably they might be doing that, right? Articular cartilage does not have no, right? So articular cartilage gets nourishments from the bone. Okay, so from the bone, there are no tiny canals in articular cartilage. That is only seen in some of the costal cartilages, right? So articular cartilage is avascular basically, and it gets nourished by the underlying bone, which is highly vascular. Right? Yeah, explain and describe, that's what I just told you. Describe is just giving an account of something, right? So if you're told describe the skin, okay, describe the skin. So you say the skin has 3 layers. It has epithelium, epidermis, dermis, and hypodermis. Epidermis is like this, dermis is like this, like that. If you say explain, it will be something like, let's say, okay, so we'll say a fracture of the femur bleeds profusely. Profusely means a lot. Okay, so that's an explain question. That is, explain why a fracture of a long bone, we will say, bleeds profusely. Okay, or that kind of question. Then you will say bones are highly vascular. When there is a fracture, the vessels inside the bone and surrounding the bone will get damaged, will get broken, and blood will come out. And that is the reason why long bones, when they are fractured, will bleed profusely. Okay, so that is explain. Explain is you're telling you're telling the examiner why something happens. Okay. Essay, oh yeah, sorry. I'm sorry, I forgot that, right? I will send you the questions, right? I'll try to do it today. I don't have them at home, that's the problem. But anyway, I'll do it. Okay, so point form of paras, the general thing is when you're writing an essay, you can write, you can add small subheadings, right? But write a little bit extra. Just don't write one sentence. If you had like if you say simple glands, you can have that as a subheading, and underneath that you write small paragraphs, right? Because it's an essay. It is okay. Don't write, I mean, writing from one end to the other in a huge sort of, without breaking down, it's difficult for the examiner, right? However, because it's an essay, try to, it's good to have a point so that you will not miss something, but make it a little longer, little explanatory, so that it looks more like an essay, right? But for those things, we don't cut marks, but you will get more marks if you write like that, right? Okay, past papers, yes, I'll work on that. Serous demilunes cover both acina and tubular. It is in mixed ones, right? So you see serous demilunes in acini, which have both acina and tubular components. Okay. Difference between lacuna and territorial. Yeah, territorial matrix is basically the ground substance which is surrounding each of the cartilage cells. A lacunae is actually a small space between the cell and the matrix, right? Lacunae is a space between the cell and the matrix, like a small cave for the cell to stay in, because surrounding the substance, right? Is it okay to write? Yeah, so I just explained about point form. Okay, right. So those are the questions that you've asked. If you have any more questions, you can ask him the WhatsApp. You can, there were several people asking questions, so you can do that as well, right? Okay, so then we will stop for now. So tomorrow's practical material, I will upload now, and you can go through that. And if you have any questions, you can ask. There won't be any live sessions for that. Tomorrow afternoon, you won't have any live sessions. So use that time to go through the whatever components have been uploaded to Moodle, right? So Sanjay sir must have told you, because these are two modules, you have foundation modules and limbs module. We will upload the lectures to the two modules, right? So limbs will go into the limbs module, so that you know which module deals with which component, right? So that you can study it separately for the exam. Okay, so whatever we teach for the foundation module, which will again go on throughout the term, and the limbs module also go on parallel. Okay, so whatever is expected of you for foundation module will be uploaded to that, and for the limbs module will be uploaded to the limbs section in the Moodle. Okay, right. So then we will stop now. Okay, are you all right? Any other questions? Anything you want to ask? Right, so we'll end it then.
Thank you, madam.
Thank you, madam.
Thank you.
Thank you, madam.
Thank you,
You're
madam.
welcome.
Thank you. Thank you,
Thank you,
madam.
madam.
You're welcome.
Thank you, madam.
Thank you, madam.
You're welcome.